So who would like to start? You can basically ask questions from anybody.
Professor.
Yes.
Hi. I'd like to start by asking a question to yourself, Asla. I wonder if you could expand more on what you mentioned about reductionist models and what directions you would like to take with respect to that.
Yeah, so with reductionist model, I mean that we don't induce such a complex phenotype that has, so that we have overlapping circuitries involved so heavily. One example, for example, I was really hoping that David Prince would be here. He's using this cortical undercut model. And it's very interesting because even though he hasn't really reported, there are some notes that also those animals might develop spontaneous seizures. Basically, what he only does is to cut the cortex or cause kind of the horizontal cut to layer 6 of the cortex. And that actually, in my view and experience, resembles to some extent the shearing effect that we actually see in the lateral foot percussion model. And he sees lots of plasticity changes and basically kind of generation of the ectogenic network there. But at least he hasn't really described if those animals developed, let's say, thalamic damage or hippocampal damage. Of course, thalamic damage could develop over the time based on the retrograde degeneration. But I think that these kind of models might serve some purpose when trying to answer to specific questions that avoid the increasing the noise level from other pathologic phenotypes.
Yeah, Aslam. I'm a student.
Yes.
So, well, first of all, I want to thank you for making mention to the epilepsy supplement and the work of the International League Against Epilepsy and American Epilepsy Society Joint Task Force. The work of Amy Bruce Cahal and all the other articles in the supplement could probably give an important contribution to the discussion that we are having today. And I invite everyone to take a look at them. And then my question, I don't really have a real question. The more I think about the story of comorbidities, the more I get confused. Because I think at the end, what we would agree to talk, you mentioned different kinds of comorbidities, and I think that is really important to clarify. Because when we talk about comorbidities, we talk, at least we think, about something that is concurrently occurring. But that is referring to the typical nosographical approach that we use in medicine, that is phenomenological. And so my question is, you said that gene expression does not respect the borders of diseases, and probably mechanisms in general do not do that. So one possibility would be to go back to the mechanisms, and mechanisms, if we knew mechanisms, probably we would not even talk about comorbidities. In the case of epilepsy, we are talking about syndromes. So a question is, for you and for the other speakers of this morning, where do we stop speaking about elements of a syndrome and start talking about comorbidities?
Yes, I agree. I would rather talk about elements. I don't know, I mean, you want to comment?
And is RNA sequencing going to be used to this, to look at these different elements?
Yeah, I mean, there are several tens of databases already available. You mean sequencing?
That looks at a large number of elements to try to identify.
Yes, so there are several databases at different levels, but the issue is that how do you specifically say that some change is related to epileptogenesis, particularly because we typically sample hippocampus. So why not, couldn't it be also related to the memory impairment that these animals is also having? So this is a real problem, how to find specific epileptogenesis related mechanisms. And I guess that's what you were referring.
Yeah, I don't know if this comment's pertinent or not, but after seeing Dr. Schatcher's presentation this morning, it's kind of confusing sometimes what actually is the cause of the disease or what's an epiphenomena. So it comes to the point where the elements of epilepsy sometimes are not elements at all. I mean, of the exact disease of epilepsy. So it's really hard to look at these comorbidities in terms of as if they were symptoms of the disorder that we call epilepsy, or if epilepsy is such a huge umbrella that we are fitting under that umbrella things that we shouldn't. So I'm not sure. I'm really not sure that we can distinguish an element from a non-element. And I don't know.
I think that at the end, each patient has its own syndrome. Each patient has its own epilepsy, and different situations can support, can induce specific conditions for each patient.
Yes, there was a question.
Hi, I'm a student too, so just to reinforce that.
Okay, thank you.
Professor Márcio, your lecture was about the ectogenic process and the temporal coding about that. How do you link the ectogenic process, the temporal coding, with the epileptogenic process, like Professor Haslan said?
Yeah, that's an interesting question. So the way, if I understood correctly, all that I've shown is about the ectogenic process. So it's about the seizure about to happen and not about the plastic events that make that circuit prone to seize. Okay, the way I link them both is actually the way I hope this would actually go is by applying this desynchronization process, I would expect plastic changes to actually take place that would inhibit the ictogenesis process, which means to say that you would have to apply this treatment for a while, and after that, the plastic changes that will take place will actually inhibit the seizure without the need of a continuous treatment. I have no evidence to support that, but I think that's the way it would be linked.
Yeah. One thing that's related to that that's kind of interesting is there's a poster by Ballista in Norbert's lab where they're looking at using odors to bring down seizures. And I think that's an interesting thing to try to bring this in. Now, it's not a trivial thing to do because it'll depend on the concentration and on the actual odor that's being used to make the system go in this direction rather than that direction.
Yeah, I mean, if this is pertinent or not, again, suppose you could act at specific oscillators, specific neural networks that would actually compete for signal processing inside the brain. So the whole idea would be to use external stimuli, not actually deep electrode stimulation, DBS stimulation, that could actually block the seizures. It would explain some phenomena like if the guy hears Beatles or he starts doing computations like 2, 4, 8, 16, and it starts doubling the numbers, he can actually stop his seizure. I mean, these very strange anticonvulsant methods have been reported in literature. So this, and some smells, there's this one case that this woman with epilepsy, she could smell vanilla and stop her own seizure. And it came to the point, which is quite interesting, that all she had to do is think about the smell, and she would stop the seizure. So she would recruit the signal even though the external stimuli was not present anymore. This is quite interesting.
And the other thing that seemed quite interesting from Marcius was synchronization, and trying to desynchronize the cells, because that would be something else to look at.
Yeah.
And the system is amazing. I mean, some of the synchronization of the visual system is within less than 1 millisecond. Yes. And the same thing in the olfactory quality.
Yeah, this is what they call a zero lag. Although synchronization process can be seen as a phase locking mechanism, which means that it's a fixed phase. It doesn't have to happen at once, but you're right, absolutely right. In some of these cases, you have zero lag, almost zero lag. They happen instantaneously. And this is not because of some speed of light communication, effective transmission. No, this is because A is projecting to B and B is projecting to A, and they start to synchronize and interact with the two oscillators, and then they start to synchronize almost with a zero lag discharge.
So, do you still have general questions related to comorbidities? Because then we could also go to talk-specific questions after that.
Related to that model, and we actually, to make sense of our results, we had to look at the red individuals. And when we separated them in different phenotypes, then we started to see something that was, you know, we have actually 3 subgroups, different subgroups of the same model. So you have to be able to open your mind to see that you have different animals, even though you use one only model. So, and we actually had one that it's very sad that we couldn't be able to get more animals of that subgroup in special, because that rat had a seizure that was located and it was not spreading to anywhere else that we were recording, of course, but only amygdala. So amygdala was there, grazing, spiking, and nothing else was spiking. And I actually, at first, I called that the anxious status epilepticus, because the animal looked just anxious all the time. Nothing else besides the EEG would indicate that the rat was having a status epilepticus. So, but we are not able to get more of those animals. So if we had the time to increase the number of animals, we might could be able to get like 8 animals or so, and to have a better idea of the pathology underlying that kind of epileptogenesis, right? So I think, yes, it's interesting that you talk about using models that are more, you know, restricted, but also if we could think outside the box and actually could look at more areas than only the areas that we know that are involved, open our minds to look at other things and other behaviors that are not classically described as epilepsy-like behaviors, and look at other areas of the brain, we could find many, many more things that could lead to new questions and maybe new answers.
Yeah, I certainly didn't mean to say that to move out from these complex models. I absolutely think that they are needed. But when we look at those, exactly as you were saying, you need large cohorts to really to start to see the patterns. And typically, as many of us, we have more than 15 animals. That's already a lot. But we might need 150 animals. And then the issue, as has been discussed, for example, in drug development, so also in mechanistic studies, I mean, these might become almost as laborious as human studies to get the significant statistical power, and this is, of course, the funding issue.
Yeah, can I say something about that? I think that's a really interesting question. You know, when you have so many different endophenotypes, that is a big problem. And how do we deal with it? And what I wanted to say is I come from physics, and that's from a long time ago. But I did undergraduate physics, and I still keep looking at physics. And in physics, what they do is not that there's this lab doing that experiment, and that lab doing that experiment, and that lab doing that experiment. Very often, labs in the entire world work on that experiment. And this is the kind of thing that we could probably get a lot of interesting things from if we have something that is not just a single disease that is very, very, very specific in diabetes type 1 versus TBI, where you have different things happening, which is very interesting, by the way.
Yeah. Was there another question? Students? Yes?
Hi. Professor Asla said that in pilocarpine model, parvable mean interneurons are lost about 5% only, but they are said to the role of synchronization in hippocampus and internal cortex. And some studies have shown that even though there's not a major loss in parvable mean, they are responsible for stopping seizures when you have an intervention on-demand closed loop by optogenetics protocol. And consider that piriform cortex, TBI, and epilepsy, do you think parvable mean intervention, greater attention could lead to another strategy in treatment?
Yeah, so yes, you are absolutely right. What is interesting in the TBI model, so we haven't really found anything in the piriform cortex, for example. And we have really looked. We have also looked entorhinal cortex. We see sometimes a little bit of cell loss in the dorsolateral division of the entorhinal cortex, I mean just below the rhinal fissure. But I mean the cortex under the rhinal fissure is very much intact. So, but what you basically said is absolutely right, because I should mention that we collaborated with Matthew Walker also in London. We sent rats for him, and he did slice electrophysiology, and he could show some signs of excitability in the hippocampus, in the dentate gyrus of these animals. But still we don't see seizures in these animals. So should we move to the topic-specific discussion, and let's start from Luisa. And you very nicely reviewed data from serotonin, dopamine, and GABA. And when I was listening you and thinking my own talk, so I was wondering to ask you two questions. So what is your feeling? I mean, because typically those were cortical samples in many cases. And is there any relationship of the changes related to where the focus is in the brain? And then the other thing is that is there any information about the order of appearance of those changes in those different transmitter systems relative to the course of epileptogenesis?
In the case of the patients, we are evaluating the different receptor binding. We obtain the tissue of patients with mesial temporal lobe epilepsy, sclerosis in hippocampus, and during the MRI, there are not important changes in the neocortex in that study. However, after the surgery, we evaluate if those cortices present dysplasia or no dysplasia, because it could happen, dual pathology. We try to avoid those patients with dysplasia in neocortex. And we compare with patients that present shorter duration of epilepsy induced by tumor or lesions. In that cases, both are patients with temporal lobe epilepsy for different causes. When we analyze the patients with tumor, we observe that those patients present shorter duration of epilepsy. And eventually, some patients with mesial temporal lobe epilepsy present also shorter duration of epilepsy, and some of them present no depression and no anxiety. We put together and we analyze by this kind of pathologies. Yeah?
Yeah. So,
in this case, we take the neocortex where the spiking activity is recorded during the surgery.
Yeah. Okay.
And the second question?
Yeah, so that was the temporal order. You partly kind of responded. So I mean, which one is first? I mean, it was earlier in the morning mentioned that often these some comorbidities actually appear before the seizure. So I was wondering if there is an order relative to the progression or duration of epilepsy.
I guess probably both are at the same time. a person who has depression, nobody knows if the temporal lobe presents epileptiform activity. I have seen that many changes are shared by both pathologists. And I want to see epilepsy not like an illness, just as a manifestation that something is wrong into the brain. Something that is wrong, and it's present with other alterations like anxiety and depression. It's interesting to see how many patients with depression and anxiety present seizures and vice versa. Probably it's the same target, the same different manifestations of the same change at molecular level. We don't know, but probably the best situation is to analyze the patients who present a kind of signs of depression, anxiety by EEG. And probably we could detect at the early periods of the epileptogenic process that person can develop epilepsy, or vice versa.
Yes?
So this is a question for Marcio. So the phase locking has also been postulated to be important for normal brain functions, and I wonder if you've looked to see whether different patterns of stimulation that interfere with seizure might also interfere with behaviors, maybe fear conditioning or maze running. And the reason I wonder is that you could imagine that the seizure is sort of hijacking a normal brain process and allowing it to spread, or perhaps it's unique, but you might be able to get at that. I wonder whether, A, whether it would interfere with the normal behavior process, and B, whether the same sort of patterns that interfere with it would be replicated with a normal process.
Yeah, that's a very interesting question. We did that, but we didn't use electrical stimulation to do that. What we did is we got a sound. These are the audiogenic animals, the animals that actually seize when you produce sound. So in order for us to look at the circuit that was being recruited in normal brain processing, we got a sound stimuli and kept changing the volume really fast, about 53.17 hertz, is that precise. So when you do that, you have a modulating frequency. Every single area of the brain that's doing signal processing from the auditory system would actually have that specific component in the FFT, the spectral analysis. So you can actually have the steady state evoked response. Now, when you look at the phase of that steady state evoked response, the phase is dispersed unless you do some kind of manipulation with the animal. For example, if you play the sound and give a foot shock on the animal, you do the typical Ledoux fear conditioning test, the phase locks. Only if you do that in an animal that had the stimulation paired with the sound. Okay, now if you do that in an epileptic animal, it's phase locked from start. So you're right. I mean, it seems like it's a propensity to synchronize these circuits. And in this case, in particular, this animal has this propensity in the auditory system. That's why we use this phase locking. We never tested this in other sensory systems. Would be nice. I mean, if we could do the same modulation frequency from light, for example, or from touch, it would be nice to see if in those circuits they would actually have a phase locking event. So, yeah, it's kind of an exacerbated facilitation of a sensory motor integration, if you would like to put that in terms of physiology. Yeah, exactly, that's what we think is happening. And also, if you look at the wars and the Wissorogenic rats, if you do a phase, this is actually the work that Johanna is presenting, if you do the steady state evoked response, it's almost threefold in energy, spectral energy of a normal animal. So you're completely right, yeah. This is what we are trying to prove with these kind of experiments. Actually, just to mention one other work, the Kalitzing and Lopes da Silva, they did this in patients that had visual flickering seizure, the one from Tokyo, the Pokemon thing that actually got a lot of kids seizing. So what he was looking at was, do they process differently what the steady state evoked response from the visual system? And the answer is yes. Yeah, they got the exact same thing that we got from the wars, but in the auditory system.
Other questions from the audience, students? Yes?
Good afternoon. Well, first of all, thank you very much for a nice presentation of all of you. And since you are talking about the mechanism, I was just wondering, Marcio, what are the mechanisms responsible for your electrode stimulation? So you put your electrodes in the thalamus, and you are stimulating both neurons, fibers and passage, so you're stimulating a lot of things. Probably the orientation of the electrodes might change between animals and the response might differ. And if I can extend this question to Dr. Bertrand in the morning, where you showed about the synchronicity between the thalamus and the hippocampus activity. Is it possible that the recording from the thalamus are, in fact, recording is a volume conductance from somewhere else that we're not measuring? So how sure can we be that both the recording from the thalamus and the stimulation from the thalamus are, in fact, thalamic responses?
Yeah, that's a big hole in our work. I mean, if you had to pinpoint something that's problematic about what we've shown, that's exactly it. But there's a big difference when we stimulated the thalamic structures and the amygdaloid structures in terms of how it would interpret temporal coding of electrostimulation. So there is some anatomical specificity to the ability of the network to actually decode these temporal coded electrical stimulation. So to answer your question, the right way to do this is to use optogenetics or some other mechanism that would allow me not to stimulate the passing fibers, right? Yeah, you're absolutely right. That's what we want to do. That's exactly the direction that we are going. And but we needed a proof of concept. What I'm trying to say here is that we needed to show that it is able. I don't know exactly the bits and bolts of how it's happening, but it's able to do it. And when you give a specific pattern, it recruits hippocampus. When you give another pattern, it goes to prefrontal cortex. So by temporally coding the stimulation, you can actually choose different pathways in which it would activate different structures. So to answer your question, no, we haven't gone that far in explaining how it works, but we were really excited to show that it did work. So now we have to go to some more refined technique. And yeah, if Dr. Bertrand wants to.
Yeah, maybe a little bit. Maybe I can also put my spoon to this soup because you mentioned amygdala.
Yeah, I'm really sorry. I should have asked for your permission before.
No, because I'm just trying to understand what is a pathway. Because when one gives you showed basically that when you stimulate basal nucleus of the amygdala, you can increase the threshold for pendulum tetrazo. Yeah. And although literature actually points out that you have to block the mammillothalamic or lesion the anterior thalamic nucleus bilaterally, yes, actually to increase the seizure threshold to pendulum tetrazo. And now you are doing basal amygdala stimulation.
Yes.
So how do you explain this pathway?
Okay, the sequence of events that we actually seen was the microstimulation, and then we drove this to the anti-epileptic results. So how do we explain this? We were looking for a structure that was highly plastic that could be dynamically associated to a specific circuit. So we certainly would not choose any primary input system, any primary sensory system. And the amygdaloid had exactly this high level of complexity that if it worked there, it would be quite interesting. The thalamic structures just had too many thalamofugal structures that were too diverse, so it was not a proper, how do I put it? It was not a good candidate at the time for doing exactly that. But to answer your questions, to answer that question specifically, we don't exactly know the exact mechanisms. And I would definitely say that one of the big problems for not knowing this is that we're stimulating everything. I mean, we're not even stimulating a specific neuropopulation of the basolateral amygdala. We're stimulating the whole thing. So. Well, again, we tried this as a proof of concept. This was the first time that we thought, okay, let's see if it can decode. If it can decode, let's see if it can desynchronize specific synchronized circuits. And when we did this with the fMRI study, and we were able to change the path of the evoked potentials, if you will, of what was recruited. That was a pretty good indicator that this might be very good for antiepileptic treatment.
Yeah. So Ed, you had.
All right, so to remind everybody, the question was, be sure I get it right, is that how do I know that I'm really recording from the thalamus as opposed to recording from somewhere else, correct? That's a very obnoxious but very scientifically appropriate question, and it's a real issue in terms of when you record these field potentials, are you really recording from where you think you're recording from? In fact, in the thalamus and the rat, one of the areas that we've run into, and it's a problem, is that if you look at the anatomy, you have some of the dentate gyrus and a little bit of hippocampus right on top. And we know when we do the evoked potentials that there are times when, depending on where we're stimulating, there is some of the field from the overlying dentate which shows up in the thalamus. And we can show that because we will then essentially anesthetize the overlying dentate. We will still get an evoked response, but it'll look different. Actually, it's a better looking evoked response. It gets contaminated with opposite polarity. With regard to the seizure, it's an appropriate question as well. We have a couple of indirect answers for that. None perfect, but you can accept them or not. One is that just the morphology. The things are so sharp. Usually if you're getting a far field potential coming from where they somehow blunt out a little bit, and this is not happening, and we were getting variations with attenuations and variations in this. We also know that if the electrodes are a little bit off outside the mediodorsal nucleus, even when we know we're getting seizures in the limbic system, we don't see anything. So just moving it slightly, it's still under the dentate, we don't see the same patterns. But I mean, it's an extremely important question because you can get fooled, because the thalamus is essentially a closed field. It does not project its potentials outside, whereas the dentate has lots of far field kind of potentials that it can project. And you have to be exactly certain about that. The issue of synchronization, all we can say is that we know that going both ways that they're really strong responses, and we can evoke responses monosynaptically out of the other, so there's clear. We also know that from some human data in which we're not dealing with the overlying and the thalamus is far enough away that you can get nicely synchronized seizure onset between the thalamus, the mediodorsal nucleus, and the limbic system. And if your electrodes are outside of the mediodorsal, you don't see that. So the far fields are still going to be in effect in those other areas, but you're just not seeing it. So, but it's a process of elimination. It's a question you need to ask. Am I really recording from where I think I'm recording, or is this coming from somewhere else? And especially when you're looking at field potentials, no matter whether you're doing slices or others, it's something you always have to think about. And there are oftentimes we found that there are contaminants from other areas that are being stimulated that you're recording. You think you're recording it directly from where your electrode is, but it's really coming from somewhere else. So it's a good question.
Can I just make one addendum to that? One of the most beautiful papers that I think I wrote was quoted 3 times. And that's quite disappointing because I really love that particular paper. And part of the paper is exactly trying to separate what is volume conduction in the conduction of electric fields in the conducting media and what is local, exactly very close to electrode potentials. So what we did in this sense, we recorded a 3-dimensional bipolar voltage of a specific area. So imagine it positioned in 90 degrees from each other and record all these 4 points. And you subtract everything from the middle one and you get a vector. That would be an estimation of the electric field of that specific point. If you put this in 3 or 4 points inside the brain, you can have a triangulation. And you can actually show that specific points of the EEG are not being generated close to where you are, but specific ones are. I really love that paper. I don't know why it didn't get quoted more often. I was quite non-didactic probably in writing it. But this, looking at EEG not in terms of monodimensional time series, but looking at it as you look at the vector cardiogram, in terms of vectors moving in space, that would solve that problem, that would actually show that those specific waves, part of them are being generated far away, part of them are being generated quite close.
Yes.
Thanks for a wonderful discussion. I was remembering a paper by George Bouchaki, I think it was in the 1990s, or 1999, something like that, which I think it will add something to the complexity we are talking about here. He was proposing at that time that if you look at pacemakers associated to Parkinsonian tremor and absence seizures, you are using particular thalamus nuclei. But the difference, in order to have the phenotype, like Parkinsonian tremor or absence seizure, like synchronization at 3 Hertz in patients or 6 Hertz in rats, is basically the outcome or the interaction between the inputs to those nuclei and the way the frequency is being at some point processed in the same nuclei. The phenotype is so different. I mean, we're talking about tremor from Parkinson and absence seizures, in which you have no behavior. So it's even more complex than just having the mistaken place or having a wrong side or being outside of the nucleus. You could be in the very same place, but the processing is so complicated. And talking about others is the same thing, because one other can be the trigger for a seizure, the same other could be the anticonvulsant other. So that switch mechanism is maybe the problem to be solved.
So I wanted to ask you, Diego, when you showed your human functional MRI study about the anterior piriform cortex, so I know that there are still many people in the audience who lived in epilepsy world in late 80s and experiencing the area tempesta time. I'm not sure if you are familiar with this concept of area tempesta. So that was Karen Gale who put bicuculin into the deep piriform cortex that has not really clearly anatomically defined. It was pharmacologically defined. But it was an area that very efficiently affected the ictogenesis. And it has been thought that it's part of the anterior piriform cortex. And looking at the primate brain, so that's actually very close to the area where this is a temporal frontal junction. And my question to you is that related to this olfactory stimulation and then this I7 mouse, even though you, as you said, it seemed to relate the stimulation more to ictogenesis. But do you know if those mice actually had epilepsy? And do you know if any of those, if there are any SNPs in olfactory receptors which might associate with epilepsy? And then what is there a topography in the olfactory pathways that particularly terminate to the anterior piriform area?
Yeah, that's a good question. So to what extent do they terminate in anterior? So in terms of the epilepsy, yes, I think that the publication was very clear that there was some epilepsy in a subset of mice, but it was not in every single mouse. And that I think was really interesting because they're not that different. So what was the difference between the ones that did show the seizures and the ones that didn't show the seizures? And that's not that clear. The one thing that he did that was really, really interesting, this is Riba, is he then changed the kinetics of the addition of the odor from a very fast kinetics, just bring the odor up, to a slower kinetics. And in the slow kinetics, they got very few seizures. So it was both where the signal goes, but also at how fast it goes over there. So that was really interesting. Exactly what the communication of a subset of glomeruli that respond to certain odors and the anterior piriform cortex is not that well understood, but it's something that needs to be understood. It's a good thing to find out which subset of odors are the ones that tend to give you seizure in a subset of, we would do it in mice, and try to find out what the connectivity is. There's now more understanding of the connectivity, but it's still way at the beginning. Maury published something really well done last year, and but doing a lot of it is a lot of work.
Yeah.
I got a comment that I would sort of like to make to everybody, and to get your reaction to, and sort of. But I'm going to start off with sort of this. When we talk about epilepsy, sometimes the conversations suggest that it is this monolithic, single entity with a single physiology. And I think one of these things to sort of emphasize, because it's going to get into the issue of oscillatory activity versus something else. And the comment I'm going to make is that there are some types of epilepsy in which there are clearly oscillations, such as absence, spike and wave discharges, when you do a photic stimulation and you get the system to begin to oscillate at set frequencies with certain inputs. But others, it's not clear that there's an oscillatory issue, and going more to the stimulation from an olfactory input, that somehow what's really happening is you're just driving a particular region into a prolonged depolarization, and then that sort of just spreads to other parts of the brain, somehow recruits them by whatever mechanisms. And so in terms of keeping the system busy with prevent oscillations, in terms of testing the hypothesis and making it clinically useful, is it going to be important then to say these are the oscillatory epilepsies? And then there's another set of epilepsies which really are more of a confluence of inputs to sort of drive the system over into a prolonged depolarization, which then sets the seizures off.
It's a very good question. There's this work by Guzman and another one by Imamura, and what they did is they got this hippocampal slice, and they looked at specific oscillatory processes, and the hippocampus, when they made the hippocampus seize, the slice seize. Well, anyway, if you do not cut anything, there's a very distinct pattern of epileptiform activity. But if you start slicing in cutting, for example, all the connections between the dentate gyrus and CA3 or CA3, you still have an oscillatory process. The only thing is that they do not phase lock with each other, so it changes completely the morphology of spikes. So what I think this kind of shows is both things are happening at the same time. You have areas with problems, and they also connect with other areas with problems, so you have to treat both things in order to get everything working, in order to get everything to go back to normal. So I do understand what you're saying, and I think it's, yeah, that's exactly the point, but I don't really know if these phenomena are actually disconnected when we talk about epilepsy.
I do agree, and I really like the question, because on the one hand, yes, this is a field in olfaction where when people look at the publications, they read like everything works perfectly. It works exactly the same way. Then you actually, usually in the figures, then you actually read, especially, let me just give an example. Local field potential. I've been reading about local field potential because I'm writing a paper. And then you start reading the papers, and they usually tell you, well, we've published these that do this, but there's others that do something different. And so there's differences, and that's very important for what happened with the seizures. There's a subset that with these two systems that were exactly the same genetically, they did something totally different. Now, it's very important to understand the difference between the different animals and why is it that, and I bet that some of that is going to be environmental. It's not just going to be genetic. There's going to be genetic input. There's going to be an environmental, which can be in the, you know, in the babies as well.
Well, I think that always we are trying to identify the specific places where the epileptic activity starts and how it propagates. However, we never think about those structures in the brain that control the rhythmic activity, such as cerebellum. During the 1960s, during the 60 years, many studies in the human brain were carried out, and they observed that stimulation of cerebellum reduces the seizure activity, and also the electrical stimulation of the cerebellum reduces the seizure activity. But if you want to induce epileptic focus in cerebellum is not possible. I think we have to focus on those structures that produce no epilepsy but can modulate the electrical activity. In addition, the brain stem. For example, the stimulation of the vagus nerve reduces the seizure activity and reduces the depression. But obviously, this nerve starts in the brain stem. Why don't think about to induce stimulation in those brain areas in order to reduce the seizure activity?
There was one question.
Can I just add one thing to that? Because suppose we look at each epilepsy as very different circuits, and we're trying to look at exactly what areas are wrong and how the connectivity makes it even worse. If you look at it in that sense, and you have a seizure that recruits the hypothalamus, and would actually, instead of having a motor seizure, you would just pump up a lot of vasopressin to the blood. You would diagnose this guy as hypertensive or a guy with a cardiovascular disorder. And suppose you have some different hyperexcitable and recruited circuits that function abnormally, and that would recruit the visual area, and they have an hallucination. What I'm trying to say is perhaps some of the same exact mechanisms are involved with a lot of neuropsychiatric disorders that we are calling comorbidities in some cases. Some cases, the epilepsy is a comorbidity of that disease. In some cases, it makes to an extent that we do not know if it's an element of epilepsy. But they could have at least some very basic common mechanisms. I mean, if we could identify those very basic common mechanisms, then we might be starting to try to define better what we are calling the disease itself or the disorder itself and the comorbidities. So I think this trying to come up to that minimalistic thing that they all have in common is quite the way to go, yeah.
Okay, so I would like to have the last question. Oh, there are two more questions. So, okay, so, but then first for you. You first. No, you first.
It's only me then. I would like to ask to Dr. Restrepo, you cited during your lecture a difference between the rostral and the caudal piriform cortex. So have you looked at neurogenesis in either of these areas in the piriform cortex?
How I looked at what? At the
neurogenesis in these new neurons, newborn neurons in these areas?
That's interesting. I have not. In neurogenesis, there's a lot of neurogenesis in the olfactory bulb. And how that influences the ones that, you know, the axons that go over to piriform, that has not been looked at, but it could be looked at, and we have not. Yeah, but that's an interesting question. In piriform, supposedly it's much less neurogenesis in the adult. I'm talking about the adult.
Okay, and then you have a question? What? Okay, so then you.
Hi. My question is for Professor Asla. When you talk about traumatic brain injury, you showed the classic paper showing that in severe patients, there are only 16. When you see the patient is very bad, and there's only 16% of patients that become epileptic. And you show your data with rats that none of them show seizures. My question is, you try to show the mechanism that lead to epilepsy, epileptogenesis. Why don't we look to a mechanism that prevents that a kind of mechanism, anti-epileptogenic mechanisms? Is this possible? We can look at one example that could be practical. If we assist a patient with severe traumatic brain injury, what can we do to prevent the epileptogenesis? Maybe enforce this kind of mechanism, anti-epileptogenic mechanism.
Yeah, so exactly for that reason, I mean, we have been wondering where to sample from the brain tissue to look at the epileptogenic mechanisms. And of course, one likely place would be that if we would know where typically the seizure focus develops. And that's why we are kind of going through all this trouble trying to find out where the seizure focus develops. So not to look at nice changes, but which have no association with the epileptogenesis. So the problem, and I would call it a problem, is that the seizure frequency is so slow. So you have to do extremely extensive monitoring actually to eventually diagnose epilepsy in these animals. And that's why we are looking other methodologies to kind of use as surrogate markers for later epileptogenesis, and this pendulum tetrazo susceptibility test is just one approach. But this is exactly where we are aiming at, because I mean we can profile whatever omics we want in any brain area. That costs lots of money, is lots of work, but what to make out of this data?
Can I add something to that question, because I was quite curious. If you compare different animal models like MCAO, like medial cerebral occlusion artery, and compare it to the TBI, would that affect epileptogenesis in a very different way, and how, and if you have any suggestions about it?
So we have done the MCO, and also Kelly has done lots of medial cerebral artery occlusions in rats, and they simply don't develop spontaneous seizures. We have done photothrombotic stroke, like has at Bertram, and in our hands, 19% of those animals developed epilepsy. There are reports which show a little bit higher percentage. In trauma, it depends a little bit on the model. In our hands, in that particular rat strain, we see about 50% to develop epilepsy. If we monitor more, probably a higher percentage will do so over the time. In mice, clearly there is evidence that when people do controlled cortical impact, that's like a more contusion model, gray matter model. So depending on the background, in Black 6 we see epilepsy in 10%. When people do that in the CD1 mouse, they see epilepsy in up to 60%. So there are these variables that you can try to increase epileptogenesis. Of course, then it's a big question, how much epilepsy do you want? I mean, if we think about, as he was pointing out, that even with the severe epilepsy, the civilian injury, 16% develop epilepsy. I mean, after penetrating injury in military population, up to 53% of the patients develop epilepsy. So this is an issue, how much epilepsy do you want? What is clinically relevant? And then basically what's relevant for our mechanistic studies?
Yeah, so, because by changing the model, you quoted Anna Maria Vesani, and the TBI must have a big blood-brain barrier rupture, and the other one not so much. So we're talking about a lot of immunoprocesses that happen in one model and doesn't happen as much in the other one.
Yeah, so we have looked actually together with Anna Maria, for example, in our model inflammation, of course, and blood-brain barrier damage, and we are looking at it all the time. So clear is that blood-brain barrier damage is very long-lasting. There's huge inflammation in these animals at early phase. That goes away in a few months. There might be persistent focal inflammation in certain areas. But certainly, if anything, inflammation probably is here just a regular injury response. Whether it really relates to the seizure occurrence, I don't think that there's any real evidence of that. Okay, so I think we are, are there any other questions from students? We always take those questions. If not, I would like to thank all speakers and the students. Thank you very much. And also the rest of the audience for asking your questions. This was a lively discussion. Thank you very much.